Our next speaker today is Dr. Chang Liu. Uh, Dr. Liu is professor and chancellor's fellow of biomedical engineering, chemistry, and molecular biology and biochemistry. He serves as both the director of UC Irvine's uh, synthetic biology and is the director of the Engineering and Health Institute. Chang earned his bachelor's degree in chemistry from Harvard University in 2005 before moving to Scripps for his PhD. As a Hertz Fellow in Peter Schultz's lab, his doctoral work focused on expanding the bacterial genetic code, specifically on translation of and incorporation of post-translational modifications and using GCE as a tool for evolving new protein functions. He then spent time from 2009 to 2012 as a Miller Fellow at UC Berkeley with Adam Arkin, working toward the predictable design of complex regulatory systems using RNA switches. In 2013, Chang launched his independent lab at UC Irvine, where he has built a research program at the intersection of synthetic biology, protein engineering, and molecular evolution. Chang's group is particularly well known for engineering specialized genetic systems that enable continuous, ultra-rapid mutation of user-targeted genes in vivo, which we are excited to hear more about today. These platforms, including the OrthoRep system, they've been used to evolve entirely new biomolecular functions, probe fundamental sequence function relationships, generate large evolutionary data sets for AI-based protein design, and even record transient biological events as heritable mutations to track development and disease progression. In addition to over 60 papers in high-profile journals like Nature and Science and Nature Communications, PNAS, JACS, Nature Chemical Biology, and so on, Dr. Chang has received numerous major recognitions, including the NIH Director's Transformative Research Award, the NIH New Innovator Award, the Moore Inventor Fellowship, the Beckman Young Investigator Award, and selection as a Sloan and Kavli Fellow, among many others. Dr. Liu, it is an honor to have you with us here today. Please take it away.
Okay, thank you very much. Everybody can hear me?
Yep.
Okay, great. And let me full screen this. Yeah, so it's my pleasure to be here. I'm really glad to talk to this audience, international audience, all interested in genetic code expansion. My PhD uh, was in genetic code expansion. Um, as mentioned, a lovely introduction. Thank you for that. And so it's nice to return to this subject, which we are dabbling in a bit here and there now. And the kind of entry point that gets us into GCE is through this evolutionary system that we developed called orthogonal DNA replication, or OrthoRep for short. And I'm going to present a short story on how we are using it to evolve aminoacyl-tRNA synthetases to support genetic code expansion. So to start, I just want to introduce this kind of major platform and concept that we have in the lab, um, major as a fraction of the work we do, which is called orthogonal DNA replication. So in the bottom left here, you can see my pointer, right?
Yes, we can. Yep.
Okay, great. So in the bottom left here, we have a cell that, of course, has a normal replication system, right? Indicating that it has a normal replication system. And what we do with orthogonal replication is we add another replication system into the cell, largely consisting of a DNA polymerase and a plasmid, that we call orthogonal to the host replication. And what this means for us is that the DNA polymerase here does not touch the genome. It's not responsible for any genomic maintenance or replication. And likewise, the genomic DNA polymerases or the nuclear DNA polymerases or the host DNA polymerases don't hit our plasmid. And in this way, this plasmid is only propagated by our DNA polymerase that we've introduced. And since it's not touching the host genome, whatever engineering modifications we do to this DNA polymerase, such as making it highly error prone, will only exert their impact on this plasmid. And on this plasmid will only be genes that we're interested in, for example, evolving in this high error rate case. Okay. And the reason we set out to build this architecture and to build out the components for it is because um, of what I'll describe on this axis. So it's a bit cryptic here, but I will walk you through it. So basically, the typical mutation rate of a cellular genome is in this region here. Let's say 10 to the minus 10 substitutions per base. So this is mu that I'm indicating. So 10 to the minus 10 substitutions per base uh, in yeast, or 10 to the minus 9 in bacteria. Usually, organisms have mutation rates in this very low range. And the reason mutation rates are low is because an organism has a very large genome. And so there's a lot of stuff you can break on it. And therefore, you need to keep mutation rates low. Now, this isn't really a problem for evolution because if your organism has a lot of information on its genome, then even a low mutation rate will access adaptations, right? Because you just have lots of pieces of the genome that you could potentially evolve to satisfy some new thing, a new challenge that the environment presents. And so we're sort of okay. And so the idea is that we have a low mutation rate because the genome is large, and there's lots to break, but at the same time, we're okay to find new function because the genome is large and a low mutation rate can access many positions for mutations to source those adaptations. All right, so that's the genome case. Now, it's a bit different when we are talking about gene-sized objects, which is uh, what I'm indicating here. If you have a gene, it's much smaller than the genome, right? So on the one hand, it will have fewer spots, all else equal, to source adaptations. But on the other hand, it's small such that, there are fewer things to break. Okay. And as a result, if you're just in the mindset of a gene, you would want a higher mutation rate to drive its adaptation. And on the other hand, it can tolerate a higher mutation rate. And so these two parameter spaces for mutation rates, per base mutation rates, are different for genome scale and gene scale objects. Um, but of course, in biology, an organism is designed such that genes are all on the same genome, to ensure their cooperation and all this. And so if you are a gene, you're not really evolving that fast, even though the genome may be evolving reasonably quickly. And so there are lots of problems that we're interested in that I think the field is interested in, that are focused on gene evolution because that is what encodes, for instance, a biomolecule. And if you're a protein engineer, for instance, you may be interested in this parameter space that a typical organism does not cover. So based on the orthogonal replication architecture, right, we can now focus the right mutational speed and parameters to our gene of interest and kind of break this lock that normally prevents things in a cell, if they're encoded on a genome, from exceeding some pretty low mutation rate before the whole genome collapses. Okay? So this is the fundamental idea driving the orthogonal DNA replication system that we've developed. And so there's now an ecosystem of components, of technologies surrounding orthogonal DNA replication. In the yeast case, which is what we mostly work on, we've established a cytosolic protein-primed replication system as the core architecture for orthogonal replication. And so this thing is protein-primed, meaning the piece of DNA that we're replicating is actually covalently linked at their 5' ends to a protein called a terminal protein. And in the replication cycle, this terminal protein acts both as an origin of replication and then roughly a free copy of it acts as the primer to initiate replication. So this mechanism is quite orthogonal from how genomes are replicated and other things are replicated in the cell. In our case, we're also in the cytosol. adding an additional layer of orthogonality because we're separate from the nuclear machinery and the nuclear DNA. This architecture we established in 2014. But of course, when we started, we were playing with a DNA polymerase that had a rather low error rate. And so over the years, we've engineered many DNA polymerases that span a mutation rate somewhere around 10 to the minus 9 substitutions per base, all the way now to a DNA polymerase that has a mutation rate in the 10 to the minus 4 substitutions per base range, which is about a million times higher than the genomic mutation rate. And this 10 to the minus 4 range, if you look a little bit more into kind of the general theory guiding mutation rates tolerable and optimal for adaptation of gene-sized objects, is around where we want to be to explore sequence space for a generic gene-sized piece of information. Um, and so we can really hypermutate things durably and also in a context where selection is trying to steer their function and can maintain that information. Okay, so there are many things supporting this key part of the components, right? We have now developed multiple or mutually orthogonal systems. We can tune the expression levels of things encoded on the orthogonal system. We can control the copy number. We have now the ability to very efficiently get genes onto this system, even more efficiently than getting genes into the nucleus. So we can start evolution, for example, from very large libraries. We have switchable mutation systems that you can induce, and you can imagine kind of a nice synthetic biology or molecular biology ecosystem around this. It's also very nice to see that other groups, such as uh, Jason Chin's group and Peter Schultz's group, have recently published orthogonal replication systems where in Jason Chin's case, they also relied on this protein prime replication system as a core uh, pair for DNA replication, a polymerase and plasmid pair, but they were able to get one established and booted up in E. coli and prove that it was orthogonal, just as we had done with our system in yeast. Okay, so the organism scope is increasing for this technology. And in principle, right, this system should be portable to all cells, given that it's mechanistically orthogonal using this um, protein primed logic. Okay, so you may ask, of course, what can we do with OrthoRep? And most of our work, including what I will show next, is to use this as an engine for directed evolution. So a gene can encode a protein, maybe an RNA, but it's focused on the engineering of a particular biomolecule. And directed evolution has lots of problems that it addresses in the biomolecular engineering space. And so we can now drive those evolution experiments with OrthoRep. In addition to these applications on biomolecular engineering, though, we are also working on projects that try to anticipate how genes might evolve in nature. We're using this as kind of a laboratory for studying the rules governing adaptation, the dynamics of evolution and its relation to history, because you have this window of one mutation of one gene now that is evolving very quickly, right? And you can use sequencing and lineage tracing and phylogenetic reconstruction of what comes out to kind of test these ideas on what governs evolutionary dynamics. And in sort of the latest uh, um, phases of my lab, we've been using evolution to generate a lot of data. So you can imagine, because we are mutating genes in vivo, we don't need to make libraries. and therefore we can search evolutionary space much more aggressively than if you had to take an outcome and make a library from it and put it back into a cell every time you want to take a step in evolution, right? So this is continuous evolution, or at least continuous mutation. And if you can generate large mutational data sets that contain many mutations guided by some selective constraint, these data are very useful for training models that can learn those constraints and therefore design protein function, certainly protein structure, but increasingly protein function based on what it learns about those constraints. And of course, underlying this is this idea of mapping fitness landscapes, because those are giving us the sequence function relationships that we would like to know in a larger space than what we just observe either in nature or in the sparse evolutionary outcomes of a typical directed evolution experiment. Okay, so this is a summary of the stuff we do in the lab. Okay, so what does this have to do with genetic code expansion? Well, as I alluded to earlier, the short story I'm going to present is our use of OrthoRep to drive the evolution of aminoacyl-tRNA synthetases, which are, of course, at the core of genetic code expansion, in that this is the enzyme that we engineer to couple unnatural amino acids with tRNAs. Okay. And so naturally, of course, tRNAs use synthetases to put natural amino acids onto tRNAs. And these tRNAs then decode codons. But if we have a tRNA that doesn't decode a sense codon, in fact, the common thing we do in the genetic code expansion field, at least historically, is to use a amber stop codon, but then create a tRNA that can bind it. Then we can have this new tRNA, which isn't normally used for any translation, assigned to a unnatural amino acid, and that unnatural amino acid would get then would be translated or would be used for translation against, say, an amber stop codon, right? But in order for this to work, you have to have a synthetase that can take the unnatural amino acid and put it onto this tRNA. And you also have to have the synthetase not take any natural amino acid and put it onto this tRNA. Otherwise, you get mistranslation in the context of this non-canonical amino acid. Okay. And so if you can get all this right, then you can expand the genetic code. And people have done this over and over again in great ways and have applied this technology to many problems. But there is still this bottleneck, at least one bottleneck of engineering synthetases that can take non-canonical or unnatural amino acids. And so this is the problem we set out to solve with OrthoRep. And the way we set up the experiment, or the sort of mini platform here, is to encode synthetases on our orthogonal plasmid, of course. These are now going to be replicated by our orthogonal error-prone DNA polymerase that will mutate this heavily. Okay, at sort of these nice optimal rates, we think that will allow us to source evolutionary innovations at a speed that selection can maintain their function and can steer their function. So as we are sampling diversity here, we're going to select for synthetases that can do the following. Synthetases that can charge the tRNA with our exogenously supplied non-canonical amino acid. And the way we select for that is by having them suppress a stop codon, right? And in this selection system, we're actually using cell sorting, okay? And we like this because the way we're doing this is we are having a stop codon in between RFP and GFP. So we can sort on the ratio of GFP and RFP as a measure of how much read-through we get. And so this gives us some control over the selection as its ratio metric. And so what we do is we do selections as follows. On the right here, you can imagine a typical selection process where in a positive selection round, as we would call it, we add the non-canonical amino acid and we are looking for things, sort of cells in this gate that have a high GFP to RFP ratio, indicating that they've suppressed a lot of the stop codon. Okay. But of course, if you suppress the stop codon with a natural amino acid, you can also get here, and we want non-canonical amino acid specific synthetases. So we can also then do a negative selection where we omit the non-canonical amino acid and we sort for things that have a low GFP RFP ratio. And ultimately, what we want to get things are things that have a high ratio of ratios, right? Which we call a relative read-through factor. Um, and that is what we're going to use to guide the selection. Okay. And the advantages of doing it this way versus the more typical way of just making a library and pulling out things that work and then perhaps mutating that library again, or the outcomes of each selection round, is the same advantages that I described earlier for continuous evolution in general, which is that we can go many rounds without too much hassle because we're not in need to make libraries. And so this allows us to explore evolutionary trajectories more deeply, all else being equal. And um, again, because there's not much labor involved in the molecular biology of making libraries and transformations, we can scale this quite aggressively, and we can do this for many experiments, different non-canonical amino acids or different replicates of the same experiment to sample different evolutionary outcomes. And the depth and scale of evolutionary search is increased to doing it this way. And so as a demonstration of this, in the paper that my talk is based on, we went after, we did 8 campaigns simultaneously. So the campaigns are outlined here. Basically, this is the parent synthetase that we started with, and this is the target amino acid that we're trying to get the synthetase to take. All of these synthetases start out with low activity against the target. And here I'm showing 2 versions of the same experiment because we did it twice and we got different solutions, as I'll show you. Okay, so that's the general template. And as an example of raw data from an evolution experiment, you will see that over rounds, so this is round 1 and now this is round 4, basically these cycles of positive and negative selection, we get improvement of our population's GFP-RFP ratio in the positive sort, okay, which is what I'm showing here. We also have maintenance of the negative gate, right, when we don't add the unnatural amino acid, which I'm not showing in this plot. But then at the end of the experiment, when we think that the population has improved enough to be worth kind of trying to extract outcomes, we will take all of the DNA in our population or all of the synthetase DNA in our population, we subclone it into just a standard strain that's not mutating, but that also has the reporter, so that nothing is changing when we're picking out clones. And then we do another positive and negative sort to pull out individual clones that make up this population and are responsible for the effect we're selecting on, which is high relative read-through in the presence of unnatural amino acid. Okay. And so as you see in the summary, the clones we pick out, so we picked out 5 from each experiment, are better than the wild type in the y-axis here, which is the relative read-through fold increase in response to non-canonical amino acid. So this is the difference in amount of read-through of the reporter you get in the presence versus the absence of the non-canonical amino acid. And you see that wild type is pretty low to start in all of these cases, but then we have increases in our evolutionary outcomes. Okay, so I think there's a Q&A post, but if someone could read it to me, if it's relevant for me to stop, that would be good. Okay, And so here are some other characteristics of our evolved synthetases. Um, this is basically a replot of the of absolute RREs, not the fold increase in the presence and absence of the unnatural amino acid. But basically, you see, for instance, that the wild type synthetase here, where we evolved against this azidomethylphenylalanine, has a low relative read-through, right? And then the clones have improved that relative read-through. And in this experiment, we also used analogs of the unnatural amino acid because I think this community knows that synthetases often have a lot of promiscuity for other related unnatural amino acids. And so we are also profiling what other functions these synthetases can be used for. And it's nice to see that you can use the same synthetase to incorporate multiple different unnatural amino acids, all of which have been typically improved over the wild type efficiency. Okay. And so we've done this for multiple cases. We've done this for multiple amino acids. These are cases where we started with a M. alvis synthetase, and we're going after lysine analogs, such as ones for cross-linking. This is a photolysine with an aziridine in it, so you can use light to trigger carbene formation and cross-link. This is propargyl for click chemistry, and so the usual kind of nice chemical handles that you can now put into proteins, you can do so better with these synthetases. And then this is to cover a series of orthoderivatized tyrosines, which we're interested in for another project. Okay, so the performance of these clones are quite good. Some of them reach when you feed reasonably modest amounts of non-canonical amino acids. They reach read-through efficiencies that's approaching just read-through of a sense codon, which is gauged at 1 here. And so they follow a nice titration curve. And so this all sort of makes sense. There was a neat observation we made, which is that one of the evolutionary outcomes for one of the synthetases that dominated actually had its own amber codon in its coding region. All right, so the explanation for this is that this was selected because it forces a very low amount of read-through when you don't have the unnatural amino acid around. Because the unnatural amino acid is itself required to make the synthetase in the first place. So as a result, we have a very low rate of read-through when we don't have the non-canonical amino acid. And this is a feature we were selecting for because we're selecting for both positive and negative. So we want high positive in the presence of non-canonical amino acid and very low activity in the absence. And so these, in fact, behave in a way where we can get that higher fold change as a function of the unnatural amino acid over variants that don't have this stop codon, which evolution discovered, or this amber codon that's now recoded. Okay, so in summary, we've used OrthoRep to drive the evolution of synthetases for non-canonical amino acid incorporation. This gives us a streamlined and scalable way to generate them. We've evolved synthetases that are efficient. And in yeast, this is particularly new because the genetic code expansion machinery for yeast has lagged behind in other organisms. And then there's this nice surprise we found of an auto-regulatory strategy for making non-canonical amino acid incorporation highly dependent, and this should give us low background in the absence and low or low natural amino acid incorporation as mistranslation at those sites. Okay, so I'll end there. I was going to give you a few thoughts for the future. Maybe I'll just condense this to one slide. So one of the things that we are really reshaping all our experiments to do is to use evolution not as a direct source of outcomes that we apply, but as a way of generating big data to train models that can produce the outcomes that we desire, right? Produce the biomolecules we desire. And the advantage of that is if you have a good model and you train it well, you can imagine it generalizing to multiple problems. We could potentially have a model for designing synthetases against all sorts of unnatural amino acids and don't have to then do an experiment for every one. And the promise is that if you train with enough experimental data across a diversity of experiments and diversity of non-canonical amino acids, for instance, in this case, your model will then generalize beyond. Okay, and so this is an idea we're testing in many different contexts. And the reason we can do it is because the continuous evolution system and the orthogonal replication system allows us to get lots of evolutionary trajectories in a streamlined manner, all of which are diverged from each other, but all of which are constrained by the function that we can impose. And in all of these assays, if you can do an evolution experiment, you can also do a high throughput enrichment experiment where you take your clones and using sequencing based enrichment, for instance, with FACS binning, you can sort your synthetases by by some sort of fitness score that tells you how well they do in incorporating a particular unnatural amino acid, and that you can do in high throughput, so you get these labeled data sets to train models. And this is something that we're doing with antibodies, for example, but we are now interested in doing this with all proteins we work on. And so hopefully that will lead to kind of a new age that's fueled by protein design and ML combined with evolution to solve these problems that we are interested in. With that, let me thank those involved. Yuichi Furuhata was the lead on this project. He was previously a postdoc and currently a PI at AIST in Japan, and I'm sure he's interested in recruiting students and postdocs, so contact him if you're interested in this work. And Gordon Ricks was a previous PhD student who worked on this project with Yuichi, and James Van Deventer, who may have spoken in this GCE series, was our collaborator at Tufts, who helped us with setting up the reporters. Thank you all, and hopefully we have maybe time for one question or something like this. All right, thank you.
That was great, Chang. Thank you so much. We'll answer all the questions. People can always log off if they want, and then they can come back to the recording if they want to catch up on any of this. So that was really great. Thank you very much. As a moderator, I get to ask a question. So I'm going to there is one online, a couple online, but I'll ask a quick one. So it's fascinating to me it's cool that you can evolve enzymes to recognize new amino acids for charging tRNAs. And I can imagine like evolving, if you think about it, is changes from A to like C, that this could occur well through changes in evolution. But if you need to go through from like A to Z, is it ever beneficial to take intermediate steps in your selection of
I see.
yeah, like get to an
Yeah,
amino acid that's
yeah,
more similar to the beginning, but also to the end, and then do iterative steps?
yeah. I think that is a very good strategy for a lot of like more ambitious changes, right? You need these stepping stones along the way. And so, in fact, a little bit of history on this project. The reason we started evolving synthetases is because we're interested in evolving an enzyme that can make unnatural amino acids. That's another project that we've published on, and we're interested in making that enzyme take basically create a series of modified tyrosines. But we were unable to go just to um, to the Z.
Yeah.
The evolution experiment didn't work. So we were like, well, we need to get some way of selecting for intermediates that had different substitution patterns. And our idea was that we would use the synthetase as a biosensor for these unnatural amino acids that we were interested in synthesizing. And so that's why we developed this series on, fluoro, chloro, iodo. And now we're using this to do this sort of substrate walking on this evolution experiment. Yeah, so I think this is an important idea, and certainly evolution uses it naturally. It's also an important idea in that it might be a source of contingency in evolution. Like if you see one protein today that does a reaction, the reason it does this reaction and its kind of evolutionary flexibility may depend on like what other reactions it was evolved to do before, right? And so this historical relationship between like previous environments and the what's embedded in the kind of latent potential of a protein may be quite tight, and this is something we're also interested in.
That's yeah, very cool. It's like I think about this in ion channels and exchangers. Like ion channels are broken exchangers, or exchangers are broken ion channels.
Yeah,
Um,
yeah, definitely. Right. Yeah. So like this is something that we're also generally interested in because, a challenge of understanding evolution is that even though the phylogenetic record gives us some decent sense of history, we don't have a history of environments.
yep.
And that's just invisible to us. So anytime you want to relate environmental histories to evolutionary trajectories and outcomes, um, you're working with a lot of unknown information, and it would be great. And we're in fact trying these experiments to do it in the laboratory, where at least the principles can reveal themselves because we have control over both environment and access to history.
Very cool. All right, so there are a lot of questions. So I'll get to it.
Okay. Yeah, and I can stay as long as
Yeah,
uh, you know.
great. Thank you very much. Appreciate that. So, can you read these questions? It might be easier for you to
Yeah,
just read.
let me read through them.
Yeah.
Okay, so the first one is by Philip Ilievsky. Hope I said that right. So there are multiple parts to this question. So the first is, does OrthoRep hypermutate particular gene of interest from start to stop, or the flanking ends, terminators, promoters, etc.? Good question. So it mutates everything on the plasmid. And therefore, it mutates also the 5' UTR, the promoter, as well as the coding region. What is typically encoded on this orthorep plasmid is quite minimal. It's just our gene of interest, and then we have a selection marker that we use for the upstream cloning of our gene onto the plasmid, but we don't necessarily even need to select for that. So roughly speaking, it's just the gene and the expression components controlling that gene. And so we do often see promoter mutations, but those typically like saturate in terms of just getting to a level of expression where it doesn't matter anymore. And so after that, or in tandem with that, all of the focus of evolution is on the gene. We can also get some mutations in these regions that the terminal proteins are attached to. But what's nice is one, these regions are very short, so they're about, somewhere around 100 bases. And the second thing is if you mutate them in a way that, for example, prevents terminal protein prime replication, they just fall out of the equation, right? In our case, the nice thing about orthogonal replication and the reason we were focused on this architecture is because if you want to replicate your gene that you're selecting on, you have to mutate as well. The mutation is occurring as you replicate. And so if you make a mutation that then prevents replication of that plasmid from working, then you're just out of the equation. Um, versus a system where you might imagine like recruiting mutational machinery to a gene. In that kind of system, you can mutate the elements that are responsible for recruiting the mutational machinery, and it won't affect the gene's ability to be replicated, so the gene can still stick around, right? And so this is kind of a coupling we try to avoid, and is somewhat related to your question, but hopefully that answered the first part. Okay, is orthogonality 100%? As far as we can tell, basically, it's very clean. So over these multiple orders of magnitude of mutation rate difference between the genome and our plasmidsystem, we don't see an increase in the genome. And I think this is really because the protein prime replication, and kind of by design or by choice, is mechanistically distinct. And you can kind of analyze the components of replication or even polymerase breathing and convince yourself that this is the right mechanism to use. Others have used other ways of targeting mutation to certain genes that are not orthogonal replication, and I think those suffer from much higher off-target effects if you look at the data. What is the fitness cost of maintaining the system? So I do think there's a slight lower growth rate when we're trying to maintain additional components, such as multiple copies of the plasmid and the DNA polymerase, but it's really not something that is noticeable except if you try to measure it precisely. So I would say just like less than 10%, something in this range. Do you control the expression level of the system? Uh, I see. Let's say you get some sort of false negatives in case your orthorep expression level is toxic, but not necessarily your gene of interest. So I think this is related to 3. So this is also the nice thing about orthorep, which is even if the system itself exerts a fitness cost, which I already said is not high, um, if you break the system, then you break replication, and therefore you just don't have your phenotype that you're selecting on, right? So it's not a viable strategy. Um, if you had a system where replication could occur and therefore your gene could be replicated and expressed, but mutation was brought on top of it, and the mutational system to do the hypermutation had some fitness costs, you could break that and you would get a viable trade-off that I think is why some other systems are not as durable. And we're very interested in durable continuous evolution because we're very interested in getting long mutational paths for all sorts of reasons, including having the levels of diversity um, for training models. Okay, so hopefully that answers your questions. All right, Michael Lang asks, um, are you able to track evolutionary trajectory on top performing mutants? Wondering if the history can tell us how important individual mutations are. Yes, but we did not do this except in a cursory way for the synthetase evolution. But for some other experiments, we will sequence more deeply, or we will do deep sequencing at time points because we want to get sort of a phylogenetic tree of the mutations and how they build. But not in this case. We haven't done it. Philip asks, how many copies of the gene of interest do you have per cell? Is it one variant per clone, or is it many? So this is a good question. In with this synthetase, with the polymerases we're using, we're probably around a 50 copy number situation. So every cell will have 50 copies of P1. And so this is also part of the reason why at the end of the evolution experiment, we just like pull out all the DNA and put them onto, now we typically just integrate into one location on the genome or put them into a plasmid and transform them to get monoclonal readouts in a later sort. The high copy number is an interesting, so in terms of characterization, it's easy to make sure you're not looking at some multi-copy phenotype where there's multiple variants contributing to some aggregate phenotype, right? So that problem is solved. The other issue is copy number will change the evolutionary dynamics in ways that are both potentially productive and potentially annoying. And so I'll leave it at that, but there's a lot of details that we've written about, and I can tell you, uh, over email or something. All right. Anton asks, did you ever get the growth-based selections to work? You briefly discussed in the paper. Yeah, thank you for asking that. So in the paper, we mentioned that our first attempt at synthetase evolution was just to use a positive negative selection based on a growth, an auxotrophy gene. A common one in yeast that we tried is ura3, right? Where ura3 synthesizes uracil, and so you need it if you don't add uracil to the cell. But then you can do a negative selection because if you add uracil, you can also add 5-FOA, which ura3 converts to a toxic nucleoside analog. And then, that can run the negative selection. So we tried that selection first, and it just kept on giving us the following. Basically, all we were doing, it seemed, was we were selecting on the copy number of the selection system. And this was because we encoded the ura3 with the stop codon in it, and some other auxotrophy markers on a plasmid. And yeast, it seems, can tolerate a very large, like somewhat heritable, but quickly changeable copy number of the selection marker itself. So in the positive selections, it was just giving us we were just selecting for tons of copies of the selection plasmid itself. And then in the negative, we were just selecting for cells that had jettisoned a lot of its copies of the selection plasmid. And so that's when we switched to this ratiometric selection. Subsequently, we have and we are using growth-based selections in a related experiment where we're using the synthetase as a biomarker for unnatural amino acids that are produced by a enzyme that we're evolving. And there we've corrected this problem where we've put the selection marker now on the genome as a single copy with a TAG in it. And so this now seems to solve the growth problem. But we have not booted it up to rerun the synthetase evolution. And so I don't know empirically if that is sufficient to solve the issue. All right, so let's see. Alexandria asks, I'm curious about the data training with random mutagenesis. How do you see the sequence space converge in terms of efficient learning? How successful do you think these will go with NCAAs in general? Yeah, so we have not done this with synthetases. And we probably won't in the near future, but maybe Ryan's group or others here might take that up. We are doing this with some other enzymes we're working on, as well as we have a big program for antibody engineering and design because, of course, there's a lot of therapeutic value in doing that, and it's a big ecosystem. And there, we're just getting into the retraining of, affinity-aware antibody design models. So I don't have too much success to report other than at least on like the specific cases we're training on, we can improve the model's ability to predict the binding affinity of a holdout set that it hasn't seen, right? But we're hoping that will generalize to more and more targets, and we have to do more antibodies against more antigens to get there. I want to just point out that in your question, you say data training with random mutagenesis. And I'm sure this is just shorthand, but just to make clear, right, this is random mutagenesis, but then guided by selection, which is key here because, we want to have highly dissimilar sequences, or at least clouds around highly dissimilar sequences, which you can't achieve just by random mutagenesis while still maintaining function, right? So we're essentially diverging things high degrees of sequence distance under functional constraint, which is the key to why this data that we want to use to train models is going to hopefully be very effective. And this is something that natural evolution produces all the time, right? But natural evolution doesn't give us large ortholog sets for functions we care about necessarily. And it's also going to be subject to like species specific things that it averages out. It doesn't have domain specific knowledge as clearly. It doesn't have clear labels. And so this is something we want to solve by using continuous evolution. Okay, Michael asks, do you anticipate another protein or small molecule foundation model will be needed? I see. Yeah, I think this is a good question. And I'm not expert on the small molecule protein interaction space and the computational work that's been done on that. I mean, I imagine, the same models that are being used to predict affinities of small molecule drugs and protein targets, can be, um, those modules in the models could be used for non-canonical amino acids. And if we put the data with non-canonical amino acids and synthetase sequences that take them into those models and update the weights or even retrain in whole, that might be reasonable. Yeah, so those models contain small molecule embeddings.
I
Okay.
think we'll probably cut it off there. I know this is
Okay. Sounds